The Rickettsiales, also called Ricketsias, are an order of small Alphaproteobacteria that are endosymbionts of eukaryotic cells. Some are notable pathogens, including Rickettsia, which causes a variety of diseases in humans, and Ehrlichia, which causes diseases in livestock. Another genus of well-known Rickettsiales are the Wolbachia, which infect approximately 2/3 of all arthropods and nearly all filarial nematodes. Genetic studies support the endosymbiotic theory, according to which mitochondria and related organelles developed from members of this group. The Rickettsiales are difficult to cultivate because they rely on eukaryotic host cells for their survival. Rickettsiales phylogeny. The Rickettsiales further consist of 3 known families: the Ricketsiaceae, the mitochondriaceae, and the anaplasmataceae. Most studies also support the inclusion of the Wolbachia, but one study has challenged this view. Here, the Wolbachia are the sole representatives of its own order, the Wolbachia, and as such not part of the Rickettsiales. See the schematic tree below. Other lineages, not clearly part of any family, have been described as well. Examples include Candidatus arcanobacter lacustris and Rickettsiales bacterium AC37b. Phylogenetic relationship between Rickettsiales and Pelagibacteriales, SAR11. The phylogenetic relationship between these two groups has yet to reach consensus in the scientific literature. Early reports suggested that they represented sister clades to each other. However, later studies suggested that this relationship is false and was due to a phylogenetic artifact, which artificially groups independent rich and fast-evolving lineages. Rickettsiales and Pelagibacteriales have both properties together. Upon correcting for this artifact, the Pelagibacteriales form a sister clade to the Rhizobiales, Rhodobacteriales, and Colobacteriales instead. Another study adheres to the sister relationship between the two clades. See schematic tree. In their classification, the relation between the two orders is retained in the subclass, the Ricketsiae, which include the Rickettsiales, the Pelagibacteriales, and the extinct proto-mitochondrian. Mitochondria themselves are not bacteria, but organelles. Reductive evolution. Ricketsiales genomes are undergoing reductive evolution and are typically small, generally 1/5 mbp, at rich, generally 40% GC, with a low coding density, generally 85%, and a relatively high number of pseudogenes. Reduction in genome size, %GC, and coding density and genes are generally attributed to genetic drift in Muller's ratchet. Genetic drift is enhanced in Ricketsiales genomes due to low population sizes, given their endosymbiotic nature, and frequent population bottlenecks. Similarly, Muller's ratchet is activated through the lack of recombination and horizontal gene transfer. The eukaryotic host cell is a natural barrier. References: